Welcome to this week's SciMon. I'm Jacques.
And I'm Swati of the Scientific Affairs team here at Illumina.
Did you see that they sequenced the fungus formed by the leaf-cutting ants? It's really cool to see how they co-developed.
I really thought they were termites.
Yeah, you know, you do get fungus-forming termites, but termites and ants are genetically very different. Termites belong to the Blattidae, like you want to go blat with them. And they're actually related to cockroaches. And when you look at, primitive cockroaches, you can see the family resemblance.
Ew.
Well, and then ants are social hymenoptera, so they're actually closer to bees and wasps.
Weird. You know, I guess they just share the dream to take leaves and turn it into high-quality food.
Yes, well, you know, it's clearly a case of convergent evolution. Usually, though, the termite carries its wood-digesting microbes in its gut, and the ant does not have that option. So they have quite a few advantages to living on a fungus, and you can see it in the leaf-cutting genome from Nygaard et al.
Well, what are some of the advantages to live on fungus?
Well, you know, the big thing that you don't tend to think about is that they don't have to deal with plant toxins. So they have a reduced number of detoxification genes.
That makes sense.
Yeah, it really does. And they also get plenty of amino acids from the fungus. So they even lost the ability to synthesize arginine.
You know, there's no such thing as a free lunch, so they must give something back to the fungus.
You know, that's true. And the ants are terribly polite. They do not digest the fungus enzymes or the spores, and they just poop it right back on the leaves, and that stimulates the fungus growth.
You know, how did the fungus adapt to deal with all this?
Well, the fungus is a Basidiomycete, and that became obligate about 20 to 25 million years ago. And they typically have these little inflated cells at the tip of the growing hyphae, and that normally consists of a single large vacuole. So it's this little bubble there, and they gradually enlarge in response to pruning by the ants. So there's this little back and forth going. And they grow into bundles that actually feed the ants.
Ooh, like grapes.
Oh yeah, just pretty much like it. And to Affairs et al., they used RNA-seq to analyze the gene expression in these gongolidia. And they found that they produce all the amino acids and plant-degrading enzymes that are needed by the ants.
So to cut the leaves and to cultivate the fungus would require a significant level of organization of the ants, and you know, because they're essentially running a factory there.
You know, that's very true, and including garbage removal. They have a very well-organized system, and there are 3 female castes. There are large workers, there are small workers, and then there are gynes. And gynes are queens that are still winged, but they're unmated.
Interesting. But you know, of course, they all have the same genes. So the differences are all epigenetic.
You know, Lee et al. Used strand-specific RNA-seq to determine differences in RNA editing between the castes. And they found 11,000 RNA editing sites that map to 800 genes.
Interestingly, 8 to 23% of the editing sites are conserved between subfamilies.
Yeah, and the level of editing for the same site also vary between the different castes.
It's true. It's amazing how much information you can get from genomics.
Yes, many of these mechanisms can only really be determined by sequencing, all the way from the fungus adaptations to the ant social structure.
Fortunately, we're out of time today, but thanks for tuning into our show. Please feel free to reach out to us with any questions, comments, thoughts, concerns, and any feedback. We always love hearing from you. Take care and have a great day. Bye.
Bye.